My name is Michael. I'm from Wake Forest University, and today I'm going to talk about data-centric artificial intelligence for environmental toxicology. So our motivation and background for this project, um, we mainly focus on the endocrine disrupting chemicals. So endocrine is a very important system to our human bodies, and its main function is to maintain internal homeostasis. So the data we get is from EPA, which is Environmental Protection Agency. They prioritize identifying and mitigating EDCS. And we are using data-centric approach in this project. So detecting EDCS requires reliable computational tools and high-quality data. And so we first talk about a little bit model-centric versus data-centric approaches. So model-centric versus data-centric. Model-centric is a typical machine learning techniques. It's mainly focused on the algorithms, hyperparameters, and advanced architectures. So it's mainly how we choose a better algorithm or classifiers to certain task. On the other hand, the data-centric, which we use in this project, it emphasizes on data quality, data cleaning, and data selection. In other words, we need to have a better input data with higher confidence for our task. So why data-centric approach for EDC classification? Because our EDC data can be very noisy and ambiguous. A representative high-quality data set may be more effective in classification. So our research objectives. So our main objective is to develop an evolutionary course set selection workflow that reduce the training set size while maintaining performance and the class balance and maximize recall metric. So on the figure right side, you can see, so we get the data from the EPA, and then we retrieve the chemical molecules. So after that, we do our experiment, which is the data-centric modeling. So these are 5 components in the data-centric modeling. First of all, a very common statistical techniques, which is filter rows, filter columns, remove highly correlated columns, and PCA, which is principal component analysis. So and the metric we choose is the recall, because we mainly focus on how much disruptor we are accurately detecting. And why this matter? Because fewer training samples mean less computation and then faster model training, which is crucial when computational resources are limited and misidentifying disruptors is more costly. So after we finishing the first 4 techniques, the 5th is the main point of this project, which is course set selection. So our starting point is we retrieve SMILES data and label information from the EDSP dataset, and we calculate the molecular descriptors. So over 200 descriptors were computed to represent each chemical structure numerically. And then we do the data cleaning steps as we mentioned before. So first 4 steps we did on this data. And so after we finishing the first 4 steps, we begin to the genetic algorithm. So it's a based course set selection algorithm. So what is the course set? So course set is a smaller representative subset of the original training data. So why we need to use the genetic algorithm for this? Because the genetic algorithm are good for combinational optimization, which is especially useful in subset selection problems. And it's also capable of preserving certain statistical properties like class balance. And the figure on the right side is the hyperparameters we implemented in the genetic algorithm. So the key steps in genetic algorithm is the first step is initial population generation. So in this project, we are using clustering method. So as we mentioned before, we have PCA. So we have 2 PCA columns. We are trying to cut each PCA columns into a lot of small pieces and random select each of them based on their PCA value. after we have the initial population generation, we choose minimizing false negative while maximizing recall as our fitness function. And the remaining step is the genetical operators, which is parent selection, crossover, and mutation. Then we iterate again and again. And implementation details and parameter tuning. So as you may say before, we have a lot of hyperparameters for genetic algorithm. So we do need to do a research. So we use the parallel computing, which is HPC cluster, to handle the large search space efficiently. And we find the final choosing parameters. So as you can see before, this is the final choosing hyperparameters for genetic algorithms. And the outcome is our genetic algorithm yield core sets that capturing around 15% of the original data size while remaining the class distribution. So you can see over here, the blue one is the original data size, and the orange one is the core set size. So you can see the orange one is the nearly half of the original data size. While we remain the similar percentage of label 1 level, that means we did very successfully for the core set because we preserve the class distribution. And experimental setup. So we choose 9 very common machine learning classifiers, which include k-nearest neighbors, random forest, extreme boosting, something like that. And we do the baseline. So we train on the entire data set and versus our core set after we implementing the genetic algorithm. And the metrics over here, we are trying to use several different metrics. And in this project, we finally choose the recall to be our final metrics because it's more meaningful to the chemical disruptors. And after we getting all the recall value, we do the statistical analysis, which is PRT test, to check if there's a statistical significance drop compared to the baseline. So the results, we show the models, the genetic algorithm can train on around 15% of data while remain the class distribution, and no statistically significant drop in performance. And yeah, and this is the results you can see over here. So the big table over here is the recall value for each of the classifiers. And after we do all the experiments, we do the PRT test based on the baseline. So each experiment compared the t-test to the baseline. And the small figure shows the p-value, which is has shown no significant drop in the performance. So the conclusion, the key takeaway is our data-centric methods like genetic algorithm-based core set selection can reduce the data sets while preserving performance. And our standard data cleaning or PCA did not yield a major performance improvement in recall metrics. And this approach is especially relevant when computational research or label data are limited. And further impact in streamlines EDC detection workflows and encourage more announced data engineering in environmental toxicology. And the future work, so for the future work, we probably might work on the larger data set and some advanced feature engineering. So also we might want to integration into regulatory pipelines. So right here, I want to thank to the Data Mine Research Lab from Wake Forest University and also Wake Forest University Star Travel Grant. And these are the reference. Thank you. Any questions?
I'm going to use my privileges to have the right first question.
Yeah.
Great talk.
Get this now.
I had sort of two parts to this. One part is thinking about how you might try to explain some of your work and work in collaboration with biologists, which is just a theme I'm going to, if you're not ready for that question, you should be now. But more specifically, I was also interested in the core setting and the selection of the whole data set as a baseline. Do you think that there would be room or a more apt comparison and doing like an archetypal analysis, or even comparing it to like a bootstrap or a jackknife of the data? So instead of the full information, seeing how it fluctuates subsampling, because I think that could also be, obviously the full data set's more empowered in some ways than a bootstrap or a jackknife. If you get lucky and you overrepresent underrepresented features, then perhaps it could be better. But it's a different kind of comparison that might speak a little bit to the nature of the data. So I wanted to ask about that too.
So that's a very good point. For this project, we only work on the larger data set initially, but I think we can do in the future work for your approach. And also, what was your first question?
The first question is about collaborating with biologists and trying to incorporate their workflow, or incorporating them more actively and being actively more incorporated into their work. Like how do you think that your work can help inform biologists in their work and vice versa?
So our work is mainly focused on how to cut the original data set into small pieces. So for biologists to use, so when they have the limited resource. So this is the main point for this project. Yeah. Any question?
Okay, so I have a question. Since I also work on genetic algorithm for some time, so I just curious, like how you maintain the reproducibility, like how you try to ensure like later you can, reproduce the data, like do you do anything with the random state on the model, or
we have
like you try to set it or just make it random, for the random state, or anything else, like because genetic algorithm is mostly based on something like randomization, right? Yeah. So how you ensure the reproducibility of the data? Yeah.
Okay.
Yeah.
That's a great question.
Yeah.
So we use random state in our project, and like I mentioned before, the initial population we used is from the PSA columns. So we have two PSA columns, and we cluster them into different pieces. Like we find that the PSA value in our project is around -1 to the +1. So we cut them evenly into pieces, which is the number we define, let's say 10 pieces. And so 2 PSA column will have 20 pieces. And this is stable for every iteration. And we define a random state to randomly select several pieces to form our initial population. Did that answer your question? Okay.
Any other questions? Sorry,
I still have one more question.
Please do.
Yeah, so can you tell a bit more? I'm not sure, maybe I missed some part of the algorithm. Can you tell a bit more on how you do mutation? Like I mentioned there, I saw you mentioned there are several parameters relating to the mutation, right? Can you explain a bit more?
Okay. Yeah. So the mutation stage is I think is a crucial stage in the genetic algorithm. So basically, we represent our initial population as 0101, like a binary string. So during the selection stage, it will run. There's several method in the mutation stage, and we choose is from our research. So we find this mutation type is very good for our project. And so basically it works like, so it randomly flips some of the index in the binary string, so to keep the diversity. Yeah, this is how the mutation in the genetic algorithm basically work.